We're going to speak about the amino acids, the carbohydrates, the lipids, and the nucleic acids, dehydration and hydrolysis. So we're going to start with the two processes that you already mentioned before, which are dehydration and hydrolysis. Dehydration reaction are condensation reactions as a different name. So condensation reaction involve the removal of water molecule. These reactions connect smaller molecules to form larger ones. So we start from a simple molecule. No, I'm writing. We start from a simple molecule. We end up in a large or complex molecule. They result in the creation of new macromolecules such as proteins, nucleic acids, and carbohydrates. So the reaction between, let's say, amino acids, as we know, the smallest unit of the protein is the amino acid, the smallest unit of carbs, let's say the glucose molecule, nucleic acids, they're made up of phosphate, of nucleic acid, of phosphatic group of sugar backbone, and nitrogenous bases. So we have these small units, let's say the amino acids or the glucose molecules in the carbohydrate, are bonded together with by dehydration reaction or condensation reaction. Dehydration synthesis example. So glucose molecules combine to form maltose and release water in the process. So what you should know about dehydration is that it releases H2O, and the hydrolysis, it needs H2O to occur. So peptide bonds in proteins are created through dehydration reactions. So let's speak about the maltose. So if you see here, the first glucose molecule and the second glucose molecule ending up with a hydroxyl group on the side. This hydroxyl groups on the sides, they combine together, taking out H2O outside, because if you see there's two O's, ending up with a glycosidic bond. Bonds. So these glycosidic bonds allow the production of water molecule from the glucose. One glucose plus one glucose gives maltose. Glucose is a monosaccharide, maltose is a disaccharide. We have two glucose molecules. Hydrolysis. Hydrolysis reactions are catabolic reactions. We spoke before about the catabolism and anabolism. If you remember, when we spoke about catabolism, we said catabolism is decomposition. This means that hydrolysis means also decomposition, means breaking down large molecules, ending up in a very simple small molecule. And it involves the addition of water molecules to break down larger molecules. So here, what do? We add H2O in order to break down the big molecules, as we see here in the example. These reactions are important for releasing energy and breaking down complex molecules. Hydrolysis breaks down nucleic acids into nucleotides. Proteins are broken down into amino acids through hydrolysis. So as we spoke before in the dehydration, amino acid plus amino acid gives a protein. Hydrolysis, protein plus H2O, which is water, gives amino acid. Here we broke the protein down, creating amino acid by the usage of water. If we see here, we have the maltose, we have the glycosidic bond, and we added water, and then we were able to add one H here, one H here, and um, and one oxygen on the side, ending up with two glucose molecules. So now let's make a comparison between them. Dehydration, removal of water molecules. So dehydration, we take out water molecules. Hydrolysis, addition of water molecules. One of the most important things you should know about these two reactions is water. Anything else is not important. The most important thing is this. Okay, the type of the reaction, anabolic, which is the building reaction. However, the hydrolysis is the breaking down reaction, decomposition reaction, or catabolic reaction. The purpose of dehydration is forming larger molecules. As we said, let's say amino acids, they end up together, we end up with protein. However, the hydrolysis breaking down large molecules. Result of dehydration creates a new macromolecules and hydrolysis yields smaller molecules. Examples, formation of protein, nucleic acids and carbohydrates. And however, in the hydrolysis, digestion of carbohydrates, proteins, and fats. The main role of dehydration, we spoke about it, is synthesis of macromolecules. However, the hydrolysis or the decomposition is releasing energy and molecule breakdown. So here we are releasing energy at the end of the reaction or heat, and we are adding water. Here we are releasing water and we are adding ATP. We needing energy or ATP to be able to allow the dehydration reaction to occur. Carbohydrates provide primary sources of energy and structural support. Composed of carbon, hydrogen, and oxygen atoms, classified into monosaccharides, disaccharides, and polysaccharides. So as we spoke about, monosaccharides are glucose molecules, disaccharides like maltose, like sucrose, like galactose, and polysaccharides are like cellulose, like fibers, like chitin, like what else do we have? We will speak about them later. So monosaccharides are simple sugars such as glucose, fructose, and galactose. No, I mean lactose is the sorry. So here we have glucose, fructose, galactose. Here we have maltose, sucrose, lactose. Okay. They cannot be broken into smaller sugars because they're already broken down. They are already monosaccharides, and they can be joined through dehydration reaction to form complex carbohydrates. So when we join one by one, we end up with a complex carbohydrate or a bigger one. So now we're gonna speak about the kind, like the structure of the carbohydrates and the kinds of them. So monosaccharides, they can be aldoses, they can be ketoses, they can be trioses, they can be pentoses, and they can be hexoses. Now, the word hexose means 6. Pentose, 5. Tri, 3. Aldose, ketose. So if you look in a chemical view, you can see that aldose comes from the word aldehyde. And the aldehyde formula is COH, this functional group. And the ketose means ketone. So the functional group of ketone is COR. R is the radical. So where you can add any methyl, ethyl group, and this kind of groups. So usually aldoses come in the end, on the ends, on the top end, on the end. However, ketones come from the inside. And this is the way to be able to differentiate. Why? Because on the ends, always we have a hydrogen atom. However, in the inside, we always have an R. We always have a carbon, you know? So that's why. So we have a triose is a glyceraldehyde. So we have triose, we have three carbons, one, two, three. And of course, we're gonna have an H and a hydroxyl group on each side of the carbon, since it's a monosaccharide. So you should memorize all of this figure for the IMAT. It's super important. A glucose is a six carbon molecule, 1, 2, 3, 4, 5, 6, C6, H12, okay? So you should know also something. So as we said, pentose is the ribose. Ribose is the same which is in the RNA. Okay, so it's the same exact, and it's made up of 5 carbons. So you should memorize glyceryl aldehyde is an aldose. Glyceryl aldehyde is a triose. So glyceryl aldehyde is an aldose plus triose. This you should know. Okay. Dihydroxyacetone is a ketose. Why? Because it have the functional group of ketone inside. Hexose, it's a glucose, and fructose, I guess. Fructose and the glucose. Now, if you look on the glucose, it has functional group COH. This means that it's an aldose. So glucose is a hexose and an aldose. Fructose, on the other side, have this same functional group, but in the inside of the molecule. This means that fructose is a ketose because it have RCO, or radical carbon and oxygen double bond. Let's write here a note. Okay, so glyceryl aldehyde is a triose, is an aldose. Glucose is an aldose, it's an hexose because it has 6 molecules, 6 carbons, I mean. And fructose, it's an aldose. No, it's a ketose, and it's a hexose. So all you should know are these three are the most important three of the IMAT. Of course, ribose, you should just know it's a pentose, nothing else. And yeah, that's all. I guess you won't be asked about this. If you want, you can put it as an additional information. Monosaccharides, glucose, fructose, and galactose. You don't have to study the structures, but you just have to know the differences between the structures. So I think just the structure of the fructose is different than the glucose and the galactose, and we have a hydroxyl group added in the galactose. But you won't be asked about this anyways. And yeah. So here in the glucose, you just have to know something. You might be asked about it. Is you always on the sides, you're gonna have the H opposing the OH, and the hydrogen up will be opposing the hydroxyl group down. So you might be asked only about the glucose shape and the hydrogen and the OH opposing each other's. Just to memorize it easily, just know that the things that are like H must be always opposing the hydroxyl in any way. And as we said, that the glucose is a is a hexose because it has six carbons. Plus, it's a aldose. If you see here, COH, which is here, COH. As we said, that fructose is a ketose, so um, it has a CH2. Yeah, fructose is a ketose. It will have ROH. It's gonna have a radical and the hydroxyl group together. Let's say you're gonna have carbon, a radical, and H. Okay. Um, carbon radical O. Here we're gonna have an H. ROH is it will have two R's and one O. So the ketose will be having two radicals. Okay, two radicals, and the aldose will be having one hydrogen added. So all you should know about to differentiate between aldehyde and ketone is in the functional group you're gonna have a hydrogen added in the aldehyde. However, in the ketone you're gonna have two radicals together. Galactose, we should not know the structure, so please focus on the structure of glucose, and that hydrogen must be always um, opposing the direction of the hydroxide ion or hydroxyl group. Disaccharides are formed by linking two monosaccharides. These sugars are connected by glycosidic bond through a dehydration reaction. Example of disaccharides: sucrose, lactose, and maltose. Sucrose is made up of a glucose and a fructose. Lactose is made up of a glucose and a galactose. Maltose is made up of two glucose molecules. Of course, between them, all of them, we are gonna have a glycosidic bond, and we're gonna take H2O outside the reaction. So, yeah, glycosidic linkages. Polysaccharides are composed of long chains of monosaccharides. These monosaccharides are joined by glycosidic bonds. Polysaccharides are classified into two categories: homopolysaccharides and heteropolysaccharides. Common polysaccharides include glycogen, cellulose, amylose, and amylopectin. Okay, we're gonna speak about each one of them. Glycogen, a storage form of glucose in animals with a branching structure. So it's a very complex structure, just having repetitive glucose molecules together, bonded with glycosidic bonds. Cellulose provides structural support in plant cell walls, and cellulose needs only the enzyme cellulase for it to be digested. So that's why usually cows have the cellulase enzyme, and that's why it's more easily digested by cows than humans. Amylose and amylopectin, forms of starch in plants, differing in their linkages, because one of them will be branched and one of them won't be branched. About this one, I'm gonna be speaking more in details after the break. Between the hours, I'm gonna be speaking about each one of them, and I'm gonna be giving you more notes for the IMAT about them and in what ways they might be asking the questions. Carbohydrates, organic compounds made of carbon, hydrogen, and oxygen 1-to-1 ratio. Means CN, H to N, ON. as we said, C6H12O6, this the glucose. Monosaccharides, the simplest form of carbohydrates, consisting of a single sugar molecule, is a monosaccharide. Disaccharides, two monosaccharides. Polysaccharides, complex carbohydrates made up of so many monosaccharide units. Energy source. Carbohydrates are the primary source of energy for the body. Now, some how we say researchers say that lipids have much more kilocalories than carbohydrates, and this is of course 100%. But as a primary source of energy for our body, if you saw that we spoke about the mitochondria and we use for glycolysis, we use glucose, we didn't use lipids. So that's why it's the primary source of energy. However, who gives more energy while decomposing and having the hydrolysis reactions? It's lipids. It's much more calories because I guess the carbohydrates 4 to 5, lipids used to be from 6 to 9 kilocalories per joules. Structural support. Carbohydrates contribute to the structural integrity of cells and tissues. Carbohydrates are stored as glycogen in animals and starch in plants. This is very important note. Dietary fiber. Certain carbohydrates, such as cellulose and trypsin, provide dietary fiber. Blood sugar regulation. Carbohydrates impact blood sugar levels and play a role in glucose homeostasis. Okay, so glucose, most common monosaccharide and primary source of energy in the body. It gives us ATP. Fructose, naturally occurring monosaccharide found in fruits and honey. Galactose, component of lactose, a monosaccharide present in milk. Sucrose, disaccharide composed of glucose and fructose, commonly known as a table sugar. The sugar that we use usually is sucrose. Lactose, disaccharide consisting of glucose and galactose, found in milk and dairy products. Maltose, disaccharide formed by the breaking down of starch, composed of two glucose molecules. Okay, so the difference, also the bonds, you should memorize them. These are for memorizing. So guys, starch is made up of alpha-1,4 glycosidic bonds. Glycogen is also a starch with alpha-1,4 glycosidic bonds. However, cellulose and trypsin is beta-1,4 glycosidic bonds. Starch form of glucose in plants, starch as we said. Glycogen, storage form of glucose in animals. Cellulose, it's a polysaccharide found in the cell walls of plants, and it provides rigidity of the plant. Chitin is the polysaccharide present in the exoskeleton of fungi. So alpha-1,4 is for starch and glycogen. Beta is this alpha. Beta is for cellulose and chitin. Okay, before I continue, I want to make sure everyone is okay. Oh, really? It's still until now my voice is low? How is that? Can you please tell me if it's until now? It was not now. Ah, okay, perfect. So now it's good. My voice is clear. Let me make sure. Just during this. Ah, okay. Yeah, because I was trying to enter and Sam was trying to enter, so it was a problem. What did it say? That H opposite to H is also found in galactose. Yeah, and look, usually when you go deeper in chemistry, you will know that H is always opposing OH. Why? Because when the molecule is rotating, it will be rotating in a way that the more dipole one will be so far from the hydrogen. Do they ask about tests to confirm presence of sugars? I didn't understand. Do they ask about tests? No, they don't ask about it, but there's a test which is called Fehling test to confirm presence of sugar. And there's a test for starch. I don't remember the name, but yeah, the sugar one is Fehling test if you want to know the name. Benedict. Benedict is for proteins, I guess. Okay, so just urine, okay, perfect. But you don't have to study them. These don't come with IMAT. These tests, I don't know what, they don't come. If you want to know the sugar is Fehling, that's all might be asked. Okay. So everything is perfect until now, I guess. Okay. Let's go, let's continue. So, amino acids are the building blocks of proteins. Some amino acids are essential for life, while others are not. The structure and function of proteins are determined by the specific amino acid they contain. A balanced and varied diet is necessary to provide all the different amino acids. Amino acids have specific structures, including a chiral carbon or an alpha carbon, amino group, NH2, and carboxyl group COOH. A variable side chain R that determines their unique properties. Glycine is an exception with a hydrogen atom as its side chain. Okay. I want to show something. Let me just make sure if I have it. I want to send it on the group. If I don't have it, I'll try to do it by next time. So guys, the structure of amino acids, the structure, like how is each amino acid, let's say valine, glycine, glutamic acid, the structures of them, you should not know them. So much people think that they must memorize the structures, but not even one time was asked about the structures. So with the structures, you should not memorize. The only two things you should know about them, you should memorize, like it's just two tricks might come about them, but I really need the glycine and another amino acid you must know about it. Okay, give me 1 second. I will send you on the group. I'm gonna send the paper on the group. It used to be one of my notes about these. Okay. Okay, I'm not finding. Yeah, I'm sorry guys for the confusion. Just give me 1 second because I really have to find the paper. 1 moment. Okay, guys, I'm gonna send it on the group. Okay, please open the group. I'm gonna send it on the group. If it's not clear, tell me. Okay, let me explain what's written. So, guys, about the amino acids, we're not asked about the structure. You should know that you won't be asked about the structure of no amino acid, but you should also know that glycine is an exception, and cysteine is an exception. So we have two exceptions in amino acids are glycine and cysteine. Why glycine is an exception? All amino acids have the structure of carbon, normally, hydrogen, NH2, COH, carboxyl group, and the radical. The radical changes for each amino acid. Each amino acid have the same exact things near the base. However, the radical is different. So what makes glycine something which is like, what makes glycine so special for the IMAT questions to be all about it? Is because it has, instead of the R group, instead of the alkyl group of the radical, it does not have a radical, it does have an H. So now here we have a CH2. That's why they say that not all, they might get, like they might get your question that says, okay, so all amino acids have radicals, or they might exchange radical by the word variable group. And then you might put yes, but no, it's not, because we have a glycine as an exception. We spoke before about the structure of amino acids, which is this, and we remember that we have a base here and we have an acid. So why do you think this molecule is able to be balanced? However, it's having a base and it's having an acid. It's having carboxylic acid and it's having ammonia at the same time. So what makes this amino group and the carboxyl group together able to handle each other? And the main reason is the buffer system that occurs during um, which maintains the medium in the amino acid. So that's why. Another thing that might be asked in the amino acid part is about cysteine. They might ask you which amino acid will have a disulfide bridges, and you will say cysteine. Cysteine is a kind of amino acid which have disulfide bridges. Also take this as a note. Means disulfide bridges means SH. Okay, a thiol group. So, what you should know about amino acids in general, that some of them are polar, some of them are non-polar, some of them are positively charged, like ionic, some of them are negatively charged, and some of them are non-polar aromatic. Okay, so some of them are polar, some of them are non-polar, some of them are aromatic, some of them are positively charged, some of them are negatively charged. What you also should know about the amino acids is the aromatic ones are phenylalanine, phenylalanine, like these you might be asked about them, and tryptophan and tyrosine. So these you should memorize, the aromatic ones. You should memorize that cysteine. Cysteine is have disulfide bridges for a ethyl group, which is SH. And the glycine does not have a radical, it does have an H. What you also should know about them is, okay, we have, okay, perfect. So as we said, there are some non-polar, some polar. Valine and glutamic acid. Okay, no, that's all. So please, guys, just know these notes. That's all about amino acids. You should not know more. The structures, if you want to know them, if you want to add some info about them, you can, but like you don't have to. So we have an amino group, we have a carboxyl group, R chain, and an H. Okay, I have a B in my room. Primary structure, the linear structure of amino acids in a protein. Amino acids and linear sequence. So the linear sequence of amino acids in a protein, this means peptide bonds. Okay. Secondary structure, regular folding patterns such as alpha helix and beta sheets, and these ends up with hydrogen bonds. Tertiary structure, the three-dimensional folding of a protein, crucial for its function, covalent bonds. And the quaternary structure have peptide bonds, hydrogen bonds, covalent bonds, and ionic bonds. So the quaternary structure is the functional structure of a protein. Who allows the functional, who allows the protein to end up in a quaternary structure? Which organelle in the cell? It's the Golgi and the rough endoplasmic reticulum. They work together and they try to end up with a functional structure of protein, because the protein cannot leave the cell only if it's functional. When it's not functional, it will stay inside the cell. It won't help the body in no way. So connected through hydrogen bonds, disulfide bridges, as we said in cysteine, and other interactions like peptide, covalent, and ionic. Quaternary structure, multiple protein subunits come together to form a complex, which is the functional structure. Okay. Now, any protein which we have in our body, this protein has a specific function. Each protein has a specific function. You might see receptors. Receptors are proteins, and the function of the receptors is to take the take the neurotransmitters, take all the things which are free in blood, and try to recognize according to them. Let's say the antibodies. The antibodies are also proteins, and they help in killing, making neutralization reaction with the antigen for the immune system. We have another kind of proteins like enzymes to catalyze reactions and to accelerate them. We have another type of proteins in the body, which is enzymes, receptors, antibodies, saliva, and all of these things. So these are proteins, but each one has a specific function. Now my question is, who, yeah. So my question is that this function, who allows this function to happen? I mean, which structure? One second. Okay, so guys, I saw in the IMAT a question which says that which structure gives the function. So now Google says something, but the IMAT says something else. So the answer in the IMAT says that the primary structure gives the function. Now, I don't, like there's a very huge confusion between Google and the IMAT. Let me just make sure about it because I'm gonna make sure. Just give me a moment. I don't want to say something wrong. Yeah, it's primary structure. Yeah, perfect. So yeah, it came on the IMAT a question that says that which structure of these structures gives the function. So you might be like, okay, the function, the protein is functional when it reaches the quaternary structure. So of course it's the quaternary structure, but no, it's from the beginning. The primary structure from the first from the protein before it becomes functional, it already knows what function will it make. You know. We have two kinds of proteins. I mean kinds, I mean type, you know? So we have globular proteins and we have filamentous. So globular proteins are or help only in function. Filamentous proteins help only in structure. Who can give me globular proteins? Globular proteins are red blood cells, are yeah, are like antibodies because they help in killing the antigens. I mean, killing the viruses. Okay. However, the filamentous structures are helps in the structure. So helps in the structure of the hair, helps in the structure of the skin, helps in the structure of, let's say, collagen, let's say keratin. These kind of proteins help in the structure, which are called filamentous proteins. However, globular proteins helps only in the function, like red blood cells, antibodies, IgA, IgE, IgG, whatever. So I think for proteins, let me just, I'm trying to remember all the questions I went through for like while solving. So I make sure that I said like all the tricks might come. Um, this is called the thiol group. Okay, perfect. Quaternary structure, protein found. Okay, subunits come together to form a complex found in proteins with more than one polypeptide chain, important for function, such as enzyme activity. Stabilized by various interactions between subunits. It have ionic, it have covalent, it have hydrogen, peptide, disulfide, everything, quaternary. Protein structures determines how proteins interact with other molecules. Key interactions include hydrogen bonds, disulfide bridges, van der Waals, and hydrophobic interactions. The quaternary structures enhances protein functionality. Understanding amino acids and protein structure is crucial for comprehending protein function. Primary structure is a linear sequence of amino acids. Didn't we do this? Protein 3D shape and no, it's a different one. There's somehow repetition. So it determines the protein's 3D shape and function. Amino acids are joined by peptide bonds through a dehydration reaction. Dehydration means adding them together and taking out water molecules. The primary structure is the first level of protein organization. Primary structure, condensation reaction, we remove water or dehydration. And then here the bonds will form. How the bonds form? Here the peptide bond will form. How do we take out hydrogen? So here we have an NH2, NHH, and here we have a COOH because it's the carboxyl group. So we have already OH here and an H here. They go out, they form water, and we end up with only these. So it ends up like this. This is the peptide bond using the condensation or dehydration reaction and removing H2. Secondary structure, interaction between amino acid backbones. Okay, two types of secondary structures, alpha helices and beta sheets. Alpha helices are held together by hydrogen bonds within the helix. And I want you to know, guys, that each time you see in biology the word helix, remember the DNA, as we said before, that we have hydrogen bond between the bases. So the moment you see the word helix, remember hydrogen bonds. H. Beta sheets are formed by parallel and anti-parallel strands held together by hydrogen bonds. This folding constitutes the secondary structure of the proteins. So beta and alpha helixes, both of them have hydrogen bonds. This is how they look like. This is the beta pleated sheet, and here the alpha helix. You don't have to know the structure. This is unimportant. Beta pleated sheet, examples of amino acids inside them. So here you might see the NH, the amino group. You might see the other group. Here is the peptide bond. No, here it's the amino acid, but it's making peptide bonds with the other ones. And the hydrogen bonds are these. Hydrogen bonds usually occur between hydrogen, oxygen, hydrogen, nitrogen, hydrogen, chlorine. Here hydrogen bonds occur between a hydrogen and an oxygen from the first sheet to the second sheet, from the first helix to the second helix. Secondary structure bond, you don't have to care about them, you just have to know about hydrogen bonds, alpha helix and beta pleated sheet. Look here, the beta looks like a sheet, and the helix looks like DNA. Tertiary structure, interaction between side chains, R groups help stabilize the tertiary structure of proteins. Hydrophilic side chains are typically on the outside, interacting with the aqueous environment, while hydrophobic side chains are on the inside, away from water. You should know also something that in biology and in our body, all our body, even in our cell membrane, even the cell membrane of any cell in our body, it consists of a hydrophilic side and a hydrophobic side. Any molecule which is made up of a hydrophilic side, hydrophilic means loves water, and hydrophobic means have phobia from water or fear water, or liposoluble means more soluble in lipids. So hydrophobic. Any molecule that have these two sides, this molecule is named amphipathic molecule. Always the hydrophilic side will be for the outside, and the hydrophobic always on the inside. Always hydrophobic inside. Any molecule which is amphipathic and hydrophilic, which is on the outside. Why? Because molecule means in the cell or cell membrane. Usually we have, um, yeah, because to be able to interact with any kind of water, the inside, the outside will be protecting the inside from water, you know? So any part which is hydrophobic is usually inside the molecule to the inside, and the hydrophilic part is for the outside. If we go to the mosaic, no, yeah, if you go to the cell membrane, I guess we're gonna speak about it later. Let's not go out of topic. So just remember that the cell membrane model, the name of the cell membrane model is called mosaic model. Okay, and yeah, these interactions gives the protein the three-dimensional shape, the hydrophilic side chains, which are these. Types of interaction that stabilize the tertiary structure include disulfide bridges, covalent bonds formed between cysteine, we just spoke about it, residues, hydrogen bonding, bonds between polar side chains, hydrophobic interactions, nonpolar side chains cluster together away from water. This hydrophobic. These are the molecules that hate water. So what do? They come here, they will try to cluster together, to collide together, be more close to each other, and try to go far from water. The tertiary structure determines the overall shape and the function of the protein. Tertiary structure can have ionic bonds, can have disulfide linkages, S2, they can have hydrogen bonds, they can have hydrophobic interactions. If you see here, in this part and this part, look, they are trying to get close to each other in a way. Why? Because they are hydrophobic. And if you look here also, you see on the outside of the tertiary structure, we have this, and in the inside, we have the hydrophobic. So always remember, everything else is always on the outside of the molecule. Hydrophobic should be always going to the inside of the molecule because they are scared of water to reach them. So always be protected from the outside. And they also have polypeptide backbone, which is this, made up of so much peptide bonds and amino acids. Quaternary structure. Multiple proteins coming together to form a complex can involve either identical or different proteins. These complexes can have various arrangements and shapes. The overall functional protein complex is determined by structure. Stabilization is achieved through various interactions, including hydrophobic interactions, hydrogen bonds, and salt bridges. Um, okay. What kind of protein do we have in our body which is super functional? Which is the red blood cells. Red blood cells are made up of the shape. So it's made up of beta, beta, alpha, alpha. So let's say that one of this structure, its structure is this. Okay, the red blood cell. So let's say one of these structures, the four structures we have, one of them is lost its structure. Let's say this was lost. Take it out. So what happens? The function will be lost. So always put in mind that the moment the structure is lost, ends up the function will lose. And this um, and this is like a loss of structure, which ends up in the loss of function, might end up in anemia, falciform anemia, and all other kinds of anemia. So how anemia happens? It's one of the structures of the red blood cell structure will be lost, leads to the loss of the function of red blood cell, leads to anemia, which won't help in which would be like so hard for the blood to be clotting and so much other things. I want to give an additional note, which out of topic, but I want to give it. So vitamin K helps in coagulation, coagulation of blood or blood clotting. So vitamin K helps in coagulation of blood or blood clotting. I don't know out of what this came to my mind, but I said it. So quaternary structure, it's everything together. Yeah, look, that's the hemoglobin or the blood molecule. So we have beta globin, alpha globin. This alpha, this alpha, this beta, this beta. So the moment one of the structures is removed, what happens? The function will be lost. The moment the function is lost, we will end up with anemia. Okay. Okay. So I want to make sure first everyone's okay until now. Guys, do you want any rest? Is it night now? Okay. R group of asparagine, Ketchup. Preparation of sugars? No, you don't have to know. Glycine is optically inactive. 100%, bravo. Exactly. Inactive because it does not have an active group. I think there was a question in the IMAT asking R group of asparagine. Yeah, I can give you the R group of asparagine. Okay. Look, make sure for me, just assure me that it is asking about the R groups. Try to find me the question, because if this really is, they are really asking about R groups of amino acids. I'm gonna send you the R groups of each amino acid in a summary. So just assure me in this. Primary amide, primary, okay. Insulin, albumin. Okay, I don't. Guys, are you okay with everything? Do you have any question? Everything is okay? I think you slept. Yes. Do you want any break? Do you want to sleep? Are you tired or like we can still do? Because I just want to make sure that everyone is focusing. Perfect, Isra. Bravo. Bravo. So everything is clear? Who is Sarthak? Are you a boy or a girl? Sarthak, you said something so nice, which is insulin. Insulin is a hormone, and hormones are proteins. Everything that ends with the IN, not all hormones are proteins. Some hormones are steroids, like estrogen, progesterone, and others, testosterone. These are steroid hormones made up of lipids. However, the insulin and albumin and no, insulin, um, any other hormones which ends with an IN is a protein. So yeah, proteins are super important in our body as hormones, as as receptors, as neurotransmitters receptors, as junctions also, as cycle junctions. They are so important. So everyone, everything's clear? Phosphatide do not have alpha 1 6 more. Yeah, exactly, Sayed. Yeah. Glycogen have both. Glycogen have alpha 1 4, it have alpha 1 6, it have everything. Glycogen is very complex molecule. Bravo, Sayed. Inside it's 1 4. Yeah, it's both. It's both. Take it from me, it's both. Glycogen have alpha 1 6 and alpha 1 4, both of them. It's super complex. That's why it has so much. It's made up of both of them. Okay, give me 1 minute to drink water and then I'll continue. Okay, so I want to do something. I want to explain out of the slide something. It's a kind of it's like more information about carbohydrates. So let's do this. Okay. So guys, I'm gonna try to add some information about carbohydrates. So if they ask about them, you already know. So number one, we spoke about amylose. We said amylose is a polysaccharide. We spoke about amylopectin. Yeah. Oh, Angela texting me. Okay, let me just make sure what does. Um, yeah. Okay, Angela. Okay. So Angela, they asked in the IMAT about something. They said, okay, um, they spoke in the question that the quaternary structure of the of the protein is the one that shows the function of the protein. And then after this, they ask in the question which one of these, which is like the options, the primary, secondary, tertiary, gives the function. So then the answer was the primary. Why? So the protein before it is functional, because as you know in the primary it's not functional. However, in the quaternary it is functional. So a kind of trick they were doing is they were trying to make you think that the quaternary structure is the one that gives function. But no, um, guys, I'm really confused about this question. It says primary, but in my mind, I'm not able to accept this. I think primary is structured, secondary is functional. Look, please, I have to ask someone because I really don't. I'm not sure about what I'm saying. I don't know. It says primary. Okay, let's go for primary. Although I'm not convinced. But okay, memorize it, guys. The structure that gives the function of a protein is the primary. The protein is functional on the tertiary and the quaternary. These two, the protein is functional. However, the structure that specifies what function will this protein will be doing is the primary. Let's say the protein is done. Amino acid, amino acid, amino acid, peptide bonds. Here the protein is done, okay? The sequence of amino acids is done. This is the primary structure, okay? In this primary structure, like while setting the amino acid sequence together, it tells the amino acid, okay, you will be an enzyme. Your function is an enzyme. When it reaches the tertiary structures, it functions as an enzyme. You know how? So in the primary structures, it tells, like, it gives the function. However, the tertiary, it becomes functional. This is what you should know as a trick. So proteins are only functional on the tertiary structure, but the primary structure indicates their function, gives them the function. They are not functional in the primary, don't understand wrong. They are not functional in the primary, but the primary gives them the function, specifies what function will they be doing when they are functional. I don't know if you guys understood. I really have to check the chat. I don't remember. I think it was talk medicine or the IMAT because I sat like 4 exams of them. So I don't really remember. So guys, did you understand it or Angela, it's clear? Perfect, 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 perfect. Yeah, let's continue. I'm gonna share the whiteboard. Perfect. Okay, guys. So we spoke about amylose and amylopectin. Amylose. It's not writing. Amylose and amylopectin. So both of them are polysaccharides and both of them are starch. Okay. Yeah, Angela, I cannot really hear you. I don't know why. Or maybe let me try to increase the volume. Yeah, my volume is good. I think the problem is from your microphone. So amylose, amylopectin. Amylose, as we said, it's alpha 1,4 linkages. Amylopectin is alpha 1,6 linkages. Okay, alpha 1,6. Amylose has no branches, and amylopectin with branches. Okay. Glycogen is super branched and super complex. And glycogen have alpha 1,4 and alpha 1,6 glycosidic bonds. And the glycogen, it exists in the liver and muscle fibers. Okay. When we increase the branching of a molecule, we increase the solubility. Okay, only an exception, which is amylopectin. Amylopectin has branches, but is but has low solubility. So this also might come a trick on the IMAT that, oh, we have high branching means high solubility. So of course amylopectin is like does it have a high solubility? You'll be like, okay, it has branches, so it is high solubility. No, it's an exception. It has a low solubility but high branches. Not really high. It has some branches. Um. Okay. So guys, screenshot this so I continue saying the notes. Okay. Now, how do we know? If you remember that we were speaking about glucose, and we spoke about that we have hydrogen, hydroxyl, hyd no, hydroxyl, hydrogen, and then we have, I don't remember how it was, you know. Anyways, how do we know, or how do we know if it is alpha or beta? Because we spoke about, we said that, okay, like amylopectin has the alpha 1,6 linkages, and we spoke about the beta 1,4 and other compounds, so and other polysaccharides. So how do we know if it's alpha or beta? It just depends on the aldehyde. We start from the aldehyde group and we see if they are different directions, means they are beta. This and this. If they are opposite, we are speaking about beta linkages. And hydroxyl and hydrogen, if they are same direction, means we have hydrogen, hydrogen, hydroxyl, hydroxyl, this is beta. I mean, this is alpha. Beta is different, alpha is the same. In our body, another note, in our body, we don't have an enzyme to break beta glycosidic bonds. Okay, so what else? So guys, we don't have beta glycosidic bonds, like we don't have the enzyme to break them. So that's why we ourselves, cellulose, which is made up of beta linkages, beta 1,4, and chitin, which is the cytoskeleton of fungi and insects, is made up of an acetyl beta D glycosidic bonds. What else? These beta linkages, our body is not able to break them. So our body usually only break the alpha, and that's why we have stored glucose. Um, we have stored the glucose in the liver, in the way or in the form of glycogen. So glycogen is stored in the liver in the form. Glucose is stored in the liver in the form of glycogen, and the glycogen is made up of alpha 1,4 and alpha 1,6. Alpha 1,6. Why did the body chose to store the glycogen and not cellulose or whatever in any form or starch? Because glycogen is made up of alpha, and our bodies can easily break down alpha bonds to form glucose when we don't have high glucose in blood. However, when we have high glucose in blood, what happens? the glycogen will start forming. So Sayed is asking something. Let me just make sure. Is amylose soluble in water? Also, does amylopectin have 1,4, 1,6, or only 1,6? Um, okay, so about amylose, um, amylopectin have only 1,6, not 1,4, 1,6. Okay, and amylopectin is branched. And is amylose soluble in water? I think so. I think so. I'm gonna search for it, don't worry. If we can digest cellulose and chitin, how do we get dietary fibers from them? If we can't digest cellulose and chitin, how do we get dietary fibers from them? So, you mean like we get fibers from cellulose because usually like your grandma tells you, oh, eat cucumber and eat, I don't know, green plants to get fibers. Okay, I tell you why. So So usually we don't have the cellulase enzyme to easily digest them. They are really hard to digest. But at the end, they are digested. Okay? How they are digested? Due to the huge acidity and of the gastric juice in our stomach and all the other enzymes which co-work with the gastric juice. We are able to digest them. Okay? But they take much more time than usual food to digest. Fibers is a kind of nutrient that comes out of them by absorption of blood in the small intestine. About your question, is amylose soluble in water? Okay. Amylose is water soluble, and amylopectin is insoluble in water. Okay. Okay, guys. So I'm gonna try to do something. Take screenshot of this. So as we spoke about, we said amylose has alpha 1,4, or let's say not branched, and amylopectin branched. So, Sayed, when we say not branched, this means that amylose structure is just about molecules linear. However, amylopectin have here, 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 here. So, which one do you think is easier to be breaking down the bonds, to be dissolving? Of course, the linear. Why? Because it does not need this much energy to break these bonds and then break the linear. However, amylopectin needs so much energy to break the branches and then to um, to break the linear ones. And as we spoke before, we said okay. When I spoke about solubility, I said high branches equal high solubility. And if you remember, I gave an exception that amylopectin. Okay, so you should just put in mind that high branches, high solubility only with the amylopectin, it's not soluble. Okay, so just take it as a note that amylopectin insoluble in water. And amylose, since it's not branched, linear, it's soluble. I'm just speaking about amylose and amylopectin. I'm not including all other polysaccharides. Okay, guys? Okay, now let me continue the notes. What did I say? Yeah, this we spoke about them. Yeah, let's see. So guys, let's say that we have a glucose molecule. Let's say we have 10 glucose molecules. And we want to, and they will undergo a dehydration reaction or condensation. So what will be the formula, the ending formula of all of them together? The formula of one glucose is C6H12O6. So anyone has an answer? I want the formula of 10 glucose molecules bonded together by glycosidic bond after a dehydration reaction. Let's see, Sayed. Try it, Sayed. Let's see. Okay, no one reached an answer. So, okay, let's see, Sayed. C60, H100, or 15. Almost, almost reaching, but bravo, you knew the trick. So usually it's if we add 10 glucose molecules together, it will end up as C60 at 120 or 60. But we should not take out of our mind that dehydration means loss of H2O. So when we lose H2O means that we are losing in each molecule, a molecule in each glycosidic bond, we were losing a molecule of H2O. So we're gonna lose from 120. So H2O, we should always put in mind that it was the loss of H2O is N minus 1. So it will be C60 H102 O51. Because we have two hydrogens, we're losing 2 times 10 is 20. Take 20, it will be 100. And yeah, but we said N minus 1 is the loss. So there are 10 glucose, means 10 minus 1 equals to 9. So we lost 9 H2O and not 10 H2O. Why? Because If we try to draw it, let's try to draw 10 glucose molecules. Glucose molecule, glucose molecule, glucose, glucose, glucose. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So look at them, guys. How much glycosidic bonds do we have? 1, 2, 3, 4, 5, 6, 7, 8, 9. On each glycosidic bond, we lose an H2O. On each glycosidic bond, we lose an H2O. Do we lose here? No. Do we lose here? No. So this means that we lose H2O n minus 1. Number of glucose molecules minus 1. So loss of H2O, the rule of it is number of glucose molecules minus 1. So here we lost 9 H2O and not 10. That's why it ends up in 102 and not 100. And it ends up in 51 and not 49. So did you get it? Did we get why it's wrong, Sayed? Guys, is it clear or should I explain? Perfect. Okay. Perfect, Humira. Okay. Perfect. Okay, let me change something. Let's say that how many, let me ask you something. How many, okay, let me write the question. Let's say there was a question that says after the hydrolysis means decomposition. Let's say who can answer this? After the hydrolysis of 10 glucose molecules, how many water are lost? Water molecules are lost. Let's see. Wrong. Wrong, yes, I wrong. Try to think of the trick. Wrong. Wrong, Isra. Wrong. Is it 7 glucose? No. It's 10 glucose. I asked about 10 glucose. I want to see how many water molecules are lost. Let's see. I don't think no one will know it. Okay, Angela, can you solve it? No, it's not written. I'm asking about H2O. How many water molecules are lost after the hydrolysis of 10 glucose molecules? So the answer is 0. Okay, now I want to ask you guys, why do you think 0? I gave you the answer. But why do you think 0? Bravo, exactly. 100%. So I'm saying the hydrolysis. I'm not saying the dehydration. And when we spoke about hydrolysis, we said the hydrolysis is the gain of water. It's not the loss of water. So the gain of water molecules is hydrolysis. The loss of water molecules is dehydration. You know, when you're dehydrated, you're losing water molecules. This one. So when they say how many are lost, and he's speaking about hydrolysis, it's zero molecules. You don't even have to think about it. Okay. 100%. You can still use a minus one. Why? Because um, the glucose molecules here are not circular. If it was circular, let's say that he told you this question. Look, he said, I have a chain, I have a circular chain of glucose molecules. Okay, of 10 glucose molecules. And now he's asking after the hydrolysis, how many are gained? So what do we do? What do we do here? Let's try to draw it. Glucose, 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 glucose. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So if we count here the glycosidic bonds, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So here, the number of water gained or lost will stay n, the same number of glucose molecules. Why? Because it's circular. However, when it's a linear structure of glucose sequence, so here we have to put it n minus 1. Why? Because we start in a place, we end up in a place. There's here still like there's still a part which is missing. However, when it's circular, we have the same number of glucose molecules, and glycosidic bonds means water lost or gained, because we always have to see the water, where is it lost, where is it gained, according to how many glycosidic bonds, because water is lost and gained only on the glycosidic bonds, nowhere else. Okay, so is it clear? Yes. By the way, how we say it. So it's the same according to the DNA or any other thing. Let's say sometimes, let me give you, we're still not inside the DNA and this questions, but I'm gonna say it because I might not speak about it later. And we don't really have time to say all the notes, so let me say it now. Okay. So let me tell you something also. Let's say he told us that we have a eukaryotic DNA, and what did he say also? He asked us about the hydrogen bonds. Okay. And then he said, okay, so I have a eukaryotic DNA, and he asked about the hydrogen bond. Let me give the example. Okay, by the way, I want to ask before I continue. Between the nitrogenous bases, A and T and C and G, how many hydrogen bonds do we have? Perfect, bravo. Bravo, Himera, bravo, Heidi. Perfect, bravo. Okay, so we have two hydrogen between A and T. This question came on the 2022, so it's really important to know it. And 3 hydrogen energy. Okay. So what did he say? He gave us this. and he asks you how many hydrogen bonds do I have in this eukaryotic DNA structure. So what do you guys can say? Give me an answer. Bravo. 17. Because here we have it 2, 2, 2, 2, 3, 3, 3, 2. Perfect. Okay, now I want to do a trick. And the trick says I change the word eukaryotic DNA and I put prokaryotic DNA. Okay? How many they will be? How many hydrogen bonds? No, not hydrogen. Not hydrogen, guys. Not hydrogen. It will be the same hydrogen bonds. Exactly. Say it exactly. It will be the same hydrogen. Okay, now I want to ask you, how many phosphodiester bonds do I have in a eukaryotic? Let's stay on the eukaryotic. How many phosphodiester bonds do we have in the two helices? Both of them together. 6 and 6 means 12. 12, exactly. So guys, 12, exactly. So phosphodiester is between the bases up, and then the hydrogen is between the double helix. Okay, now if I say how many phosphodiester bonds do we have in the prokaryotic DNA? Let's say that this DNA structure is a prokaryotic structure. It's not a eukaryotic structure. How many do you think? Let's see who's the smart. So Sayed said same. Does anyone else have a... Okay, we'll repeat. So I said that let's change the question in a better way. Let's say that this DNA, it's a prokaryotic DNA. How many phosphodiester bonds do we have between the two helixes, the first one and the second one, and add them together? I'm asking about phosphodiester, not hydrogen, and I'm asking it's prokaryotic DNA and not eukaryotic DNA. Yeah, think guys. Bravo, exactly. Why 14 say it? Let's see the explanation. Correct answer, but no explanation. Unacceptable. Bravo, 100%. 100%. Okay, so guys, because prokaryotic DNA is a circular DNA, so these two are connected and these two are connected. So here we added one phosphodiester and here we added one phosphodiester. Understood? Here's the trick. So that's why the word circular is so important in the question. The moment you see circular, always you have to add one. And that's why I spoke about this after I was speaking about the glucose. So always the word circular adds one when I was speaking about water molecules and now the DNA. So take this into consideration. The circular adds one O. Between the bonds, okay. 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 So if humira. Look. Let's do this. Okay, humira. Focus. So any bond we have, let's say AT, this is a DNA double helix. Let's say ATCGA and TAGCT. This is the DNA double helix, okay? The bonds between these here is called a phosphodiester bond. And the ones here okay, are called hydrogen bonds. Now, what is our trick here? What am I trying to tell you guys? That eukaryotic DNA have linear double helix, and the prokaryotic DNA have circular double helix. So the moment we saw circular, now it is linear. So how many phosphodiester do we have in eukaryotic? Now we're focusing on this. 1, 2, 3, 4, 5, 6, 7, 8. This is the phosphodiester. So here we have 8 phosphodiester. However, if it's a prokaryotic DNA, means circular DNA, means this, all of this double helix is connected because it's circular. Circular means it's connected. A here, T here, C here, G here, A here. And then on the other side, we have a T here, A here, G here, C here, and a T here. So if you count here, you're gonna be adding one phosphodiester on them. So this will be connected to this, and this will be connected to this. So when these two are connected from the sides, means we added one phosphodiester here plus one, and here we also added a one. So instead of having 4 here and 4 here, now we have a 5 here and a 5 here. So we have a 10 phosphodiester as a total for prokaryotic DNA. It's clear or not? One of the variant bonds. Yeah. Peptide bond. Okay, here. You're welcome. So peptide bond, if it's a form of covalent bond. I wanted to speak about something. I wanted to speak after this, but I'm gonna ask a question before and then I will speak about it. Just let me ask the question, Sayed, and I wanted to put it as a note also. So yeah. So the question is, which part, which organelle in our body also is like prokaryotic DNA, have a circular DNA? Mitochondria. Bravo. Okay, perfect. Okay, I'm gonna ask also something. Mitochondria, yeah, because it used to be a prokaryote before, and according to the endosymbiotic theory, it became a eukaryote, but I mean, it became able to adapt with other organelles, so that's why. Bravo, okay. Okay, let me ask you also something. Share my book. So let's say we have, I tell you which part or which answer is correct, or which one of them has circular DNA? Liver cells, um, bacteria. You solved it in the IMAT? Yeah. I think Said solved all the past papers before, and the neuron. Yeah, exactly. So which one of them? Neuron and bacteria. Okay, why do you think a neuron? So why neuron is an option? Neuron cell body is circular. Um, so it's all of them. And for you said the neuron cell body is circular because you mean like you have here the dendrite, and here the cell body. Oh, you have a point. You have a point. But I think a neuron is a cell, is a normal cell. And I think liver cells are normal cells. And these two cells, because they are normal cells, they have mitochondria. So that's why we consider that they have circular DNA. Why? Because it's inside the mitochondria, and these cells have organelle, which is the mitochondria. This is how I thought about it. But yeah, you may have a point. I don't know. But like I think any kind of cell in our body have the mitochondria. This means, of course, it has a circular DNA. Bacteria because it's a prokaryote. Um, I want to ask also. I want to say a note. I also saw it as a question that insulin. I mean, it asks about the neuron. It says that does the neuron have insulin allele or gene? What do you think, guys? The neuron have nothing to do with glucose or with insulin. But do you think it does? Okay, why Sayed? Why do you think it does? So its answer is all of these, yeah. Every cell except has a circular DNA then. 100%. It was a question I forgot why. Okay. Yeah, you just know how to solve. Okay, guys, does neuron have insulin allele? Yeah, it does. Why? Because all of our neuron cell, يعني. Why? Because all of our cells in our body have the DNA. Okay, it has a nucleus. Neuron has a nucleus. And the nucleus contains our DNA. And all our cells hold all the genes of our body, hold all the alleles of our body. Not even one cell does not hold an allele of our body. The insulin one, the one of glucagon, the one of keratin, the one of color of the hair, the one of color of the eyes, all of the cells, all of our body cells, all the liver cells, all of the kind of cells that we have in our body hold all the genes or the alleles because they all hold the same chromosome. Like they hold the genetic material because all of them have cells. Each cell of us hold our DNA. Okay? The only difference is that this allele is functional. Let's say the insulin allele, where is it functional? Where do you think? In which type of cells the insulin allele is functional? Let's see if you know. Pancreas, 100% because the pancreas produces insulin and glucagon as hormone. So yeah, guys. So iris of Langerhans, exactly. So all our cells of our body have all the alleles, have all the genes, have all the genetic material, but each one of this genetic material, each specific one, let's say the keratin one, where is it in the hair? Okay, so the hair part, the keratin um, allele about the hair structure is only functions in the hair, you know? And the same for insulin, only functions in the pancreas, but it does exist in the neuron. It does exist in the liver cell, but it functions in the pancreas cell. So in this way, you should take into consideration genetic material because it is all over our body, all over our all mutated cells. Not mutated, mitotic cells. Okay. Every cell except red blood cell has circular DNA 100%. Red blood cell does not have a nucleus. Red blood cell does not have mitochondria. Okay. Now, guys, do you want to break? Do you want I'm okay to, if you want, we can. Let me see. Is peptide bond a form of covalent bond? Yeah, yeah. Remind me, thank you. Okay, let's speak first about the glycosidic bond. We have two glucose molecules, let's say this and this. Here between them, we have a covalent bond. And we have a covalent bond normally, you know, because they are sharing electrons at the end, the elements inside them. And these from down are glycosidic. Same goes for peptide for two amino acids together. So we're gonna have from N to C, from the amine group to the carboxylic group. Take this as a note that the direction of the buffer system inside an amino acid is from amine group to the carboxylic group, from N to C. So we were speaking about the peptide bonds. We will have here N, the amine group, and NH2, let's say, and this. Here we will take out an H2O, and we will end up with a peptide bond. So nitrogen and carbon will be sharing electrons. So yeah, it is a covalent bond. Glycosidic amino acid. No, I was speaking in this about glucose, two glucose molecules. This is maltose, and this is amino acid. Okay. So I want to ask guys, do you want us to continue the slides, or you're tired, you want to sleep, or what do you want? Tell me if everything is clear, if you have any questions, if you want more notes, or if you want to continue the slides, or tell me what do you want. What do you think is the best to do now? Because I think everyone's sleeping and I don't want sleeping. Okay. Humira, Humira, Ifra, Angela, go ahead. No break. Break or no break?